All right, everybody, we are back with chapter two, and this time we're going to talk about the chemical basis of life. So, if you think back to chapter one and our levels of organization, you should remember that our simplest and smallest level was the chemical level. So, in this chapter, we're going to focus on that chemical level that combines to create all other living things. And so, I want to remind you that even though our chemical level was that simplest and smallest level, our cellular level, the second level, is our first level of sustaining life. So everything that we're going to talk about in this chapter is super important because it's our smallest building blocks. But keep in mind that until we get our full form of a cell, then we are not necessarily sustaining life yet. We're just building the pieces right now. In chapter three, that's when we'll move on to our cellular level and focus on one single cell and all the components there. So I just wanted to make that distinction. So, the chemical basis of life, we're going to talk about a lot of chemistry concepts, and this certainly is not a chemistry class, but without understanding some of the basic chemistry concepts, we can't move forward on the anatomy and physiology side of things. So, we're not going to get super in depth into a lot of this stuff. Hopefully, for those of you that have had some chemistry experience previously, a lot of this will just be a review and a reminder. And let's get into it. There we go. Okay. So this table shows all the common elements in the human body, and you can see our top four here, especially hydrogen and oxygen, make up a large percentage of the number of atoms that are in the human body. If we look at it by weight, you can see that oxygen and carbon are by far the highest percentage. And then some of these other ones, even though we don't find a whole lot of them, are still super important, especially things like sodium and potassium and chlorine and calcium. We'll talk about those throughout our different chapters for sure. But right now, you really need to remember that these are our most abundant elements. That's what this slide is obviously highlighting here. All right, so we just said that those four at the top, hydrogen, oxygen, carbon, and nitrogen, are the four most common atoms. So what is an atom? So an atom is made up of a nucleus, that's center dot here, surrounded by an electron cloud. So the only thing that we find in the electron cloud, I bet you can guess, is electrons. If we were to zoom in on our nucleus here, what we find are protons and neutrons. And so each of these three elements, or what we call subatomic particles, have their own charge to them. So electron is going to have a negative charge, so one electron, one negative charge. A proton, you'll see we can also abbreviate as an H plus. H plus is hydrogen. Hydrogen is made up of just one proton. So if we have one proton by itself, we can still say that's the same as a hydrogen ion because it's got that charge. And then neutrons, we put this N0, and this is just to denote that it's not charged. Now, oftentimes when we start looking at actual molecules or other atoms, we won't necessarily use the zero as a common marking to denote that there's no charge. You'll definitely always see a positive and a negative if there's a charge for an atom or an ion or molecule, whatever we're talking about. But typically, if you don't see the positive or the negative, we just assume that zero would be there. So that if you don't see plus or minus, we know it's a neutral atom. Okay, so a true element is made up of all three of those components, and the way we define elements is by the number of protons. So another word for the number of protons within an element is the atomic number. So if you take a look at the periodic table, 1 through 100 something is all based on how many protons those elements have. So hydrogen has one proton, so its atomic number is 1. Carbon has 6 protons, so its atomic number is 6. And so we define all of our elements. These are our top four, but we define all of them based on numbers of protons. You may have heard the word isotope before. And so that is, for example, if we used carbon, carbon has six protons and six neutrons and six electrons. It's usually a neutral element, six, six, and six. Let's say we find a carbon atom that instead of six protons, or sorry, instead of six electrons, has 10 electrons. It's still going to be carbon because it's got six protons. That's how we define it. But we consider it an isotope because it has a different number of electrons. And so that's going to make it a different charge and it's going to behave differently, even though it's still a carbon atom. And then the other thing to point out here, we use this planetary model when we look at atomic structure. So, like I said, if you've had high school chemistry and you guys have talked about electron clouds, this may look a little bit familiar to you. Sometimes these are called shelves or levels of the cloud, and you'll see that only a certain amount of electrons can go in each level. So for our inner ring, you can see we can only have two max. You see we have two here, four here. That gives us our six electrons for carbon. Even though oxygen has eight electrons, we still only see two within our inner shell. And then we have six around the outer shell to give us our eight. And part of the reason that we organize them into these levels, and we'll talk about this a little bit, is that this is how our different elements can share or steal or borrow electrons. And that's how we can make bonds and attachments between the elements to form molecules and eventually really big structures. Okay, I mentioned this or sorry, not number, but word briefly earlier. And so an ion is what we use if we have an unequal number of electrons and protons. So this is when we actually get what we call a net charge. And so that can, of course, be negative if we have more electrons than protons, or it may be positive if we end up with more protons than electrons. So for our example here, we have sodium chloride, our end product here. This is table salt. And so, of course, that's made up of a bond between sodium and chlorine. And so if we look at sodium here, we have our two electrons on our inner shell, our eight on the outer shell or middle shelf, and then we have one lone electron hanging out here. One other thing that's kind of nice to know about electron clouds is they like to have full shells. So that means this one guy hanging out here all the way on the outside, he kind of feels left out. He's not really having a good time out here. So sodium is going to want to give away that electron so that it can have these nice full shelves again. Now let's look at chlorine over on this left side. You can see we have our two inners, our eight in the middle, and then we have seven out here on the outer shelf. So it's only missing one to get that complete outer level. So it would make sense in this case that sodium has one kind of extra electron. Chloride's missing one from the outer shelf. So chlorine is going to pull that electron so that it has a full outer level. Sodium's going to give theirs away because now they have full levels. And so now, our chloride just got an extra electron. Remember, electrons are a negative charge. So now we have one more electron than proton, and we're going to have that negative charge. Sodium just gave away an electron, so it now has one more proton than electron. And so now we can give it this positive charge. And you see, we started with sodium atom and chlorine atom. Once that charge comes into play, now we call it the sodium ion and the chloride ion. So here's some other examples. We talked about the hydrogen ion as kind of a special example because that's really just one proton by itself with no electrons. Our sodium ion is going to be positive because sodium wants to give away that outer electron. Potassium, very similar situation. And I'm sorry for the typo here. This should be a K plus, not an Na. I will fix that in your course slides. Chloride ion. So we just saw chlorine has that one gap in their outer shell, so it tends to want to pull that in. And then calcium. So this is our first example that we've seen with a 2 plus, and so that just means we have two extra protons compared to electrons. Same thing if we saw 2 minus. That means it stole or borrowed two electrons. So now it has two more electrons than protons. So it's not always just going to be a plus or a minus. We can have ions that have a charge of 2 plus, and they can even go higher than that. Okay, we're going to talk briefly about solutions because this is going to be important when we actually talk about transport within the body. So we're going to stick with our same example, NaCl. So we saw our chlorine or our chloride can get that electron from sodium. It's a really strong bond here because they're actually completely swapping an electron. Now, if we were to put this cube of our sodium chloride, our salt, into this water, they're going to split into their ionic compounds. So all of these red and blue structures are representing water molecules. So this is your oxygen in red, and then our two hydrogens in blue. So H2O, two hydrogens, one oxygen. And so one thing that I do want to point out quickly here is that we have these kind of weird looking images here with a plus and a minus. This symbol here means that it's a little bit positive and a little bit negative. So for water molecules, our hydrogens are a little positive, our oxygens are a little negative. So it's not quite like this example where sodium donates a whole electron to chloride. In this case, our hydrogens and oxygens kind of share an electron, just not equally. So think oxygen's bigger. It's going to be stronger, so it's going to pull the electron closer to it. That means it's going to be partly negative because it's got a little bit more of an electron. Our hydrogens are going to be partly positive because that electron's farther away from them now. So they're not quite a full positive charge like our sodium ion was, but they're more positive than when they're by themselves. So we can talk about that a little bit more in class or shoot me an email if that concept's a little confusing. I just wanted to point that out so you guys know what's going on here. And so what you can see is that our partially negative oxygens are attracted to those positive sodium ions, and our partially positive hydrogens are attracted to our negative chloride. So when we have two ions that are combined, when we put them into water, put them into a solution, they're going to separate out into their ionic components. A couple of kind of just vocab words that we should talk about when it comes to solutions so that we're all on the same page. The water itself, or the liquid in a solution, we call the solvent. The solvent. Whatever we are going to dissolve into is the solvent. This salt cube in our example here is the solute. So whatever our solid or material that we're putting into the liquid is the solute. So in our case here, we get a solution between the solute of our salt cube and the solvent, which is water. And it's important to point this out. If we put this NaCl into something else, a different liquid that had other chemical components, this isn't necessarily going to happen because hydrogen bonds, this partial positive and negative, is a really important kind of concept or base of why solutions do what they do. We're not going to get into depth on why that happens. Like I said, this isn't chemistry, but keep in mind that this doesn't just happen whether we drop it into soda or something else. Okay. The real reason we're introducing solutions is to talk a little bit more about acids and bases. So before I jump into this stuff, let's just take a look at our chart and make sure that we understand what we consider an acid, what we consider a base, and how we actually give those a rating. So the pH scale, this pH really means percentage of hydrogens. And so we'll talk about that a little bit more. That'll make sense once we go over this example. And so it's from 0 to 14. So if it's from 0 to 14, we know seven's right in the middle. And so you definitely need to know this 0 to 14 number. You don't need to memorize these tens down here, but what you should understand is that when we're all the way down on this end, we have a low concentration of sodium. I'm sorry, I'll backtrack. Low concentration of hydrogen ions, high concentration of hydrogen ions up here where all of our strong acids are. So 7 is dead center. If you follow that over, you see distilled water. That just means pure water. That's going to be a perfect 7.0. It's not acidic, it's not basic. If we go above 7, then we get into our basic or alkaline solutions. So alkaline or alkalinity is another word for something that's basic, something that has a pH greater than 7. Here recently, there's been a big uptick or trend over the last year or so in alkaline water. You can find it in almost every single grocery store that has a higher pH than our body. So the idea is that it can kind of try to help neutralize any acids that are within the body. So our acids are going to go the other direction. So the closer to 0 we get, the stronger the acid. So if I follow this all the way over, I see that hydrochloric acid has a pH of 0. That's as strong of an acid as we can possibly have. And so you can also see, like I said earlier, this is where we have our highest concentration of hydrogen ions. And so HCl, this hydrochloric acid, is considered such a strong acid because if we put that into water, it's going to completely what we call dissociate. They're going to completely separate. We're going to get a lot of hydrogen ions, a lot of chloride ions. So the percentage of hydrogen ions is going to be really high. So solutions with proton concentrations greater than water are considered acidic. So remember, this hydrogen ion is the same thing as saying one proton because our hydrogens have one proton, one electron. When they separate into an ion, we just have the H plus, just the proton. So sometimes we can say that acids are H plus donors or proton donors because they want to dissociate and give away those hydrogens. So the main thing for you guys to keep in mind is this scale itself. You should know your 0 to 14. Know that distilled water at 7.0 is dead center. And then know the closer to 0 we get, the stronger the acid. And that's because it's going to have more dissociation. All the H and Cl's are going to separate. And so it's not always going to be HCl. So you may have different acids, but there's almost always going to be hydrogens because we can't donate hydrogens without the presence of hydrogen. The other thing that I want to point out here is our normal pH for blood. So the dead center kind of perfect point for blood is 7.4. So our blood is a little bit alkaline compared to distilled water. The normal range, if you remember that back from our homeostasis talk, we can't always keep it dead center, would be 7.35 to 7.45. And so that's definitely good to know, especially for those of you that are kind of going into the healthcare field, especially you nurses. When a blood gas is taken, that tells us how much oxygen and carbon dioxide and bicarbonate and that kind of stuff is in the body. One of the other numbers we'll get is the blood pH, because we have to keep that within check for our systems to function normally. And we'll talk about this again in our respiratory chapter. Okay, before I jump to buffers, let me just make sure. I want to point out up here that our highest base is sodium hydroxide. So the strongest acid we have is hydrochloric acid. The strongest base is sodium hydroxide all the way up here. Okay, so we just talked about this normal pH of our blood being around 7.4, between 7.35 and 7.45. We have a lot of different systems in the body that help us to keep our pH in check. And so you'll hear as we go through our body systems, multiple systems that we'll talk about that have this responsibility. One of the most important buffers in the body is the bicarbonate ion. And so its job is, since it's a negative ion, to bind with these positive protons, these H plus ions, and now we have a neutral ion. Sorry, not an ion anymore, but a neutral molecule. And so buffers help us to get rid of too much H plus or too much OH negative if we're talking about the basic side of things. And without these buffers, it would be a lot harder for us to maintain our pH because anytime we introduce something acidic or alkaline, there would be a much bigger effect on the body. So our buffers help us maintain that middle. So if you think of the bowling alley, your buffers are the bumpers that you put up when we're going to go too far one way or the other. We have a buffer that tries to help us out and keep us in the lane. All right, talking about chemical reactions. So we're going to go through a few different kinds of reactions. A lot of times, the name of the type of reaction pretty much tells you what's happening. So the first one we're going to talk about is dehydration synthesis. And so synthesis is your key word here because this is when our enzyme is going to take monomers, or another word for monomers would be building blocks. They're going to bond them together to form polymers or chains of those building blocks. We're taking bricks and we're building a wall. Hydrolysis is kind of the opposite. This time our enzyme is taking the polymer, we're taking our wall, and we're breaking it down into individual bricks. And most of the time, we're not taking a sledgehammer and knocking it down and tearing our bricks apart. We're going to take it apart so we can reuse those bricks to build the next wall that we need. So typically in our reactions, we're not trying to be wasteful. We're often going to recycle any of those monomers that we can so that we can make new polymers. So think in your heads for just a second. If we're looking at the right side of the screen, think which one of these makes more sense to pair with this image. Okay, if you said dehydration synthesis, good job, because we have two component parts that are forming one bigger structure. So we know that's a synthesis reaction. If we had AB being broken down so that we have A and B again, we reversed this order, then we would have hydrolysis. And so a couple important things that I want to point out about enzymes here. So this is our enzyme, and you can see it's just like a puzzle piece. So it's shaped perfectly on this side for molecule A to attach. It's shaped perfectly on this side for molecule B to attach. So our enzymes are going to be very specific for the reaction we're doing. They're going to be, this enzyme is purely for AB to AB. So it's got to be the right enzyme. This is the way the body can make sure that there's not molecule C and D bonding to this enzyme when it shouldn't be. So it's going to fit perfect like a puzzle piece. It's got to be the right shape for the right molecules. And so molecule A binds with the enzyme, molecule B binds with the enzyme, and then it kind of squishes it together. A little more complicated than that, but as far as we know, we're making that bond. The other main point that I wanted to point out is if we look down here, our enzyme at the bottom looks exactly the same as our enzyme at the top. So our enzymes are typically going to be recycled as well. We're not adding the enzyme into the molecule, it's just helping that chemical reaction happen. So anytime that we make a bond, it's going to take energy to do that. It takes us energy to build something. When we break a bond, that energy is going to be released. So making a bond is going to take energy. Breaking a bond is going to make or release energy, is really the better term because it's not brand new energy. So keep that in mind because that's going to come up again as we get a little farther along. Okay, you guys certainly do not need to memorize our boxes over here, but what you should understand is this second part, or really in this case, the first part and second part of our words. So dehydration synthesis. We said this was a synthesis reaction because we're going from A, B to AB, two building blocks into a bigger structure. So that was the synthesis part, putting two things together. Dehydration is the way that this actually happens. And like I said, I don't need you to memorize this, but I do need you to remember that a dehydration synthesis reaction is going to produce water. Produce water. And so it can be kind of counterintuitive because we call it dehydration. But if you're thinking we're taking the water out of the pieces to build the big structure. So we have two amino acids. These are building blocks for proteins. To put those two together into a dipeptide, into two pieces that are connected, we have to have our big block and we also produce water. Dehydration synthesis. Oops, sorry, guys. Hydrolysis literally means water destruction. So it's going to consume water. So anytime you see the word lysis or this suffix lysis, you know we're talking about destroying something. So this time we have our larger structure first, and we want to break it back down into building blocks. And this time, instead of a protein, we have a disaccharide. And we'll talk about the building blocks of our different macromolecules in a little bit. But disaccharide means two, disaccharide sugars, two sugars. And we want to take that disaccharide and break it into two monosaccharides. So we have glucose, which is what our body can use as a sugar. This time, for us to be able to break that bond, we're going to consume water. So this time, water is on the reactants portion of our reaction or our solution. And then we have the H and OH. You can see where that water goes on the product side. So dehydration synthesis, putting two building blocks together, it's going to produce water as well. Hydrolysis, when we're breaking these apart, we need water to do that. It's going to consume water. All right, now we're going to get into our macromolecules, and you'll see that almost all of our macromolecules have this hydrocarbon ring structure. And so all of these are considered organic macromolecules, meaning that we have both carbon and hydrogen. And so we call these hydrocarbons or these hydrocarbon rings. That's often how our structures tend to organize themselves. You guys do not need to be able to produce these or know like glucose is like if I took this word away, I wouldn't expect you to look at this and know it's glucose and fructose. So our building blocks of carbohydrates, our monomers, are glucose, or the main example in the body, I should say, is glucose, because fructose is a monosaccharide that is found in fruits. So this is the sugar you would find in a fruit. So these are both individual units. So by themselves, they're monosaccharides, one sugar unit. If we have that dehydration synthesis reaction and put those one glucose plus one fructose together, we get a disaccharide, two sugar units called sucrose. So that's what's happening here. One monosaccharide bonded creates a disaccharide. Now, once we get to three or more, we then call it a polysaccharide, just meaning many. So if I had three glucose molecules put together, I would call it a polysaccharide. If I had 3,000 glucose molecules chained together, I would call it a polysaccharide. And the main polysaccharide or polymer in the body is glycogen. So this is how we can store glucose. So these are a bunch of glucose chains put together. So we call that a polymer or a polysaccharide. And so glycogen is going to be found mostly in our liver as well as our skeletal muscles. And when we don't have enough available glucose, the body uses the stores to break off individual pieces. And so that way we have usable sugar again, because our body can't just use up this big glycogen chain. It's a more efficient way to store it, but it's not usable by our cells. We have to get it back to that monomer glucose for that to work. All right. The next thing we'll talk about is nucleotides. And so nucleotides we associate with DNA and RNA, but there's also a really important nucleotide monomer called ATP, adenosine triphosphate. And so if we look at just this part, kind of ignore the yellow for now, you'll see this in DNA and RNA. So this is what we call a nucleotide or a base pair. For ATP, we have three phosphate groups. So adenosine is this blue and gray portion. If we add two phosphates, we call it adenosine di-2-phosphate, but we have three, so adenosine triphosphate, ATP. If you know what ATP actually stands for, it tells you exactly what the structure is. And so this is our energy storage molecule. And if you think back from a few minutes ago, I said when we were talking about enzymes that when we make a bond, it's going to take energy. So enzymes help us use a lot less energy, but it still takes energy. So if you think when we make bonds, we store that energy in the bond. So in this case, we have two bonds because we have three phosphates. And so the way our cells use this energy is that when we need that energy to be released, we're going to break off one of these groups. When we break that bond, energy is now available. It's released from the bond, and we can use that for whatever process our cell is trying to do at that point. So our cells are constantly making new ATP molecules to store energy. And then when they need it, we can break off our bonds, and that releases energy that can be used by the cell. Okay, now we'll talk quickly about DNA and RNA. So like I said, look at our blue structure here. So this is that adenosine structure we were just looking at. And so we call these nucleotides our base pairs. And so you'll see in our ladder structure here that's twisted, we always have the same color. So red and blue are always together, green and yellow are always together. So we have these nucleotides. We have A, G, C, and T. So those are in DNA, A, G, C, and T. So we had our adenosine goes with thymine. And so, oops, sorry. And then for our G and C, we have our guanine always goes with cytosine. So that pair always happens. We always get the same matchup. In RNA, we swap out. We won't find this T. So our T turns to a U, uracil, it's called in RNA. So that's one difference there. So this kind of illustrates that difference or what we would see. So DNA and RNA are basically instructions for creating proteins. It's basically our recipe for creating everything in the body. So if we're looking at a DNA sequence, if we wanted to follow these nucleotides, we'd say ATGGTAC. So that would tell us to make this protein. In RNA, we swap our T and U. So you see it looks almost identical, except for every time a T pops up, we're going to put a U there instead. The other big difference between DNA and RNA is that DNA double is a double helix. Deoxyribose nucleic acid. It's got two sugars. RNA is just a single strand. So this is representative of DNA. It's that latter kind of structure, what we call the double helix. RNA would just be one single line, one piece. And so our DNA and RNA, their main job is to be responsible for our genetic material, and they use that genetic material to produce new proteins. Carbohydrates that we talked about on this slide, we said we had our monosaccharide glucose, our polysaccharide glycogen, and we need glucose to make ATP. So it's really carbohydrates are how we get energy. So we can make ATP using glucose and oxygen. So carbs are your big energy source. Nucleic acids are going to be our genetic information. All right, a couple more macromolecules to touch on. Our next one is proteins. So that's the major category there. And our building blocks are going to be amino acids. Amino acids. So there's actually 20 different amino acids. And so these are just some examples of specific amino acid monomers. Remember, whenever you see that mono, you know you're talking about just one simple building block. And the way our amino acids work is that we kind of put a couple together with what's called peptide bonds, and then that keeps, sorry guys, I keep bumping my mouse. And so our peptide bonds, we keep adding pieces and pieces and pieces, and eventually we get a long chain of them. Eventually we get more like a sheet that keeps folding and folding, and then we eventually get this structure that has thousands and thousands of amino acids. So proteins we could consider our most important macromolecule. We need all of them. We can't function without any of them, but proteins tend to be the structure and functional portions of everything in the body. So if we don't have enough proteins, if proteins start to fail, then everything else in the body is going to start to fail. So that's why we tend to consider proteins our most important macromolecule. Now, out of these 20 different amino acids, about half, really nine of them, or um, we can actually make enough of ourselves. So we call those non-essential. Even if we didn't eat any protein, we'd have enough to use within our own body. The other ones, the other half, are our essential amino acids. And so that means we don't have the capability to make enough of them or recycle enough of them within our own body that we have to eat them to have enough to function. So that's why you really have to have protein in your diet because we need all 20 of these amino acids for our different proteins. And so if you ever see something advertised as a complete protein food, most animal products are complete proteins. Some non-traditional protein sources that are complete proteins are something like quinoa, which is really more of a grain, but it has all the different amino acids. A complete protein food is going to have all 20. So complete protein means you're getting all 20 amino acids, but it's really about half of those that we have to eat in our diet to have enough. So for proteins, you're thinking structure of everything else in the body. So that's kind of your big keyword for the category is structure. Okay, our last category here is lipids. And so if we're using our kind of layman's terms, lipids are another term for fats. And our main dietary fat is going to be in the form of triglycerides. And so triglycerides are made up of our three fatty acid building blocks, our three fatty acid tails connected to our one glycerol tower here. So three fatty acids, One glycerol. And so, like I said, this is most of our dietary fats. This is another one that we associate with energy. So when you're sitting, just resting, most of the time, your body is actually burning more fat than it is carbs. But when we say our primary energy source or what we can use to make energy quickly, you should be thinking carbohydrates and namely glucose. Think about a marathon. When those people are running in a marathon, they often will have, whether it's fruit snacks or gel, that has more or less pure sugar in them. You don't see people popping fat pills when they're running a race because that's not going to do you any good. It's going to be our longer term, slower burn energy. And the other big thing is insulation and protection. It helps us maintain our heat. And then think of why our fat goes towards our abdomen. Our skull does a great job of protecting our brain. We have our vertebrae to protect the spinal cord. We have our rib cage to protect the heart and lungs. And then we said in our abdominal cavity, that's where all our other organs are. So they don't have any bony protection, which is part of the reason why fat tends to go to that area to provide a little extra insulation. So if you see energy, your first thought should be carbs if you just see energy. If you see energy plus insulation plus protection, then we're talking about fats. Okay. Another category of lipids that we'll actually talk about a bit more in depth in our next chapter is phospholipids. So this will kind of just be an introduction to this structure, and we'll talk about it in the cell membrane next chapter. So our phospholipids are broken basically into two halves. We have the head, and this is considered polar. So this is our phosphate head. And so polar is another way of saying it has a charge. And then we have our nonpolar, our non-charged region of our two fatty acid tails. So more or less, this is a triglyceride that lost its third tail and gained a head. So the other important introduction piece for this is to understand that our polar head is hydrophilic. Philic means to love, so it loves water. Our polar, our charged head region doesn't mind interacting with water. Our nonpolar tail here is considered hydrophobic. So think of a phobia as a fear. So it does not like to interact with water. In general, two things that are polar tend to like to interact. Two things that are non-polar get along pretty well. But if we try to put something that's polar through something that's non-polar, it doesn't work so well. So remember, water had our partial charges on the oxygen and hydrogens, so it's going to be considered polar or partly charged, which is why it gets along well with this head region. The tail region has no charge, so it doesn't like having to interact with that partially charged water. And like I said, besides just kind of remembering the head and tail and the hydrophilic hydrophobic matchup right now, don't worry too much about these because we're going to talk about this specifically with the cell membrane. All right, our last structure for our lipids, we're going to talk about cholesterol. And so most of the time when you hear the word cholesterol in daily life, it's not a good thing. So you'll see a lot of commercials for the medication class statins. And so statins are the class of drugs that lower our cholesterol. And really, when we're talking about lowering our cholesterol, we're talking about lowering the quote unquote bad cholesterol. And I'll hit on that in just a second. But cholesterol is a super important lipid for the structure of many, many different things. So here's just a few examples. Estrogen and testosterone are primary sex hormones that are responsible for a lot of different development in the male and female systems, rely on cholesterol for their structure. Bile salts that we use within our digestive system. So you guys don't need to necessarily know these specific examples, but it's really just to highlight how important the presence of cholesterol is. And so some of them help with membrane fluidity. You'll see again, when we look in chapter 3 at our cell membrane, we'll see some pieces of cholesterol along with our phospholipids that are helping give us structure. And it also can be modified to produce what we call steroid hormones. So not all hormones are steroids, but a lot of hormones are considered steroids. And anytime we see this word steroid, we're not thinking of the shoot it up, get big and strong type of steroids. But you should think we need cholesterol to produce that. So kind of circling back to how I started this slide, we have good cholesterol and we have bad cholesterol. So when you see commercials about lowering your cholesterol, really what they're trying to lower is our VLDL cholesterol and our LDL cholesterol. Those stand for very low density lipoproteins and low density lipoproteins. Those are kind of like big blobs of fat that just float around. They get stuck on the walls of our arteries. They get stuck in the heart. They just clog things up. That's what we don't want. That means that's when we have too much cholesterol that we're not organizing it very well. What we do want is enough cholesterol to build structures like this. And we also have a form of cholesterol called HDL. That's high density lipoprotein. And you can think of HDL like a wrecking ball or a bowling ball. It's tightly packed, so as it's going through our system, it does a good job of actually breaking up some of that low density stuff and collecting junk that's not supposed to be there along the way. So you guys aren't going to be tested on that VLDL versus LDL versus HDL, but because it's something that we hear so commonly talked about in terms of our society's health picture, I think it's important to understand what that means. All right, so I know that was a lot, guys. Like I said, this intro chapter can be a little bit tricky because we're going over a lot of our basic concepts. And I will see you for our last chapter of this unit, chapter 3, where we will zoom out just a tiny bit and start looking at the actual structures that make up an individual cell. Thanks for listening.